and get started then. Welcome to the ASA Biopharmaceutical Section's quarterly safety working group webinar. My name is Susan Mayo. I'm a senior mathematical statistician at FDA, and I co-lead this with Michelle Zhang, who's an executive director at Stealth Biotherapeutics. Today, we are happy to have joining us Dr. Veronica Pei. She is the Lieutenant Commander in the U.S. Public Health Service and a lead physician on the Associate Director for Biomedical Informatics in the Center for Drugs. Before we get started on that, I wanted to also introduce you to the rest of the panel and say just a little bit about the working group. So the panel will include Mike Fries, Jürgen Kubler, and Zhao Ni. They are head of biostats at CSL Behring, owner of Quantitative Scientific, and head of the DMD biostats at Sarepta Therapeutics. And then we'll have all speakers and panelists with the ability for the audience to provide any questions you have for them to respond to with a brief conclusion. A slide we standardly provide in our webinars is to just say a brief amount about our working group. It has 3 work streams. Work stream 1 is about interdisciplinary safety evaluation. Work stream 2 is about safety monitoring and statistical methodology. Work stream 3 is about real-world evidence and bridging that with clinical trials for safety decision-making. And then on the side, we have a communication and outreach group of which this webinar is a part. So today's presentation from Veronica relates to two of the elements of work stream 1 and also work stream 2 in our working group. That's why those are highlighted in red. So that's all I have at this point. I wanted to then introduce, I'll stop sharing my screen, Veronica, and you can go ahead and start yours.
Okay.
Thank you. I just wanted to introduce Dr. Pei. She's a commissioned officer, as noted in the Public Health Service, and serves as lead physician for the Biomedical Informatics and Regulatory Review Science, or BERS team, in the Office of New Drugs at FDA. She's a board certified emergency medicine. She's board certified in emergency medicine and clinical informatics. In her current role, Dr. Pei serves as subject matter expert on numerous committees and working groups related to data standards, business informatics, and governance, and is involved in development, implementation, and support of bioinformatics initiatives within OND, including the standard safety tables and figures. And she's also FDA's topic leader for ICH M11 expert working group on the structure and content of clinical protocols. So thank you, Veronica, for your presentation today, and over to you.
Okay, thank you, Susan, and thank you for having me here today to talk to you guys about a data challenge issue with specifically in DILI. And I want to go off video just in case there are connection problems, but can you see my slide?
No, still Susan's.
Oh, maybe Susan, you have to stop sharing.
Sorry about that.
That's okay. Okay, let me try this one. Screen.
I can see Veronica.
Okay.
Share. Oh, wait, is it not? Can you, hold on a second. Can you see my screen or no?
It was there and it just disappeared.
It's coming. It's coming. There it is.
Okay.
There we have it. Thank you.
Okay, perfect. All right, so let me go off of video and I'm gonna pull up my other. So I should have my, all right. Everybody can see my slide. So I know many of you, but for those of you who don't know me, I started the FDA as a clinical reviewer on the liver team. And then I was a team leader on the liver team. So in my career at the FDA, at least half the time I spent having to deal with livers. And so when I transitioned into informatics, I still sort of hold issues related to DILI near and dear to my heart. So helped to write led the guidance development for the NASH technical specifications, which helps to submit data to help sponsors to guide them on submitting data for MASH. And now included in that piece of it is really drug-induced liver injury. How do you submit data for that? So today I'm going to talk about a little bit about the challenges that we see from a regulatory agency when we see data submitted for DILI, and then how what sort of resources industry can leverage to address some of those issues. So here's my standard disclaimer. I don't have any financial disclosure, and this is my opinion only and does not represent official policy of the FDA. So just a review about DILI. We know that it's uncommon, but it is potentially lethal, right? And it is one of the reasons why a lot of drugs fail to achieve marketing authorization, or they're withdrawn early in the program, because if there's even a hint of DILI, sponsors may not want to spend the resources into developing this drug. Oh, I'm hearing some background noise. I'm wondering if people can mute your mics, please. Thanks. And then it's one of the major reasons for post-marketing restrictions. So for PMR, PMC, and then it's really difficult to diagnose during the drug development phase. So by the time we actually get the data submitted for marketing application, often it's too late to go back and try to gather additional data. So this is an opportunity for us to sort of communicate with industry on some of the challenges that we see in the data that we get. So really, I sort of try to categorize the data challenges into different categories. And really, there are lots of challenges intrinsic to just assessing DILI, because there's really a very broad range of liver-related data, right? So it's not just laboratory, it's not just AE. And even within the laboratory, we talk about whether labs are gathered in central versus local laboratory, or even on unscheduled study visits, how to report that data, or whether that data is even captured. And then the liver-related data are really found in different data sets. So it's not always seen in the ADAE, and the abnormal labs are sometimes reported in the ADLB, although we often see a lot of issues with missing data. And I'll go into that in more depth. And then even with those subjects identified as potential Hy's Law candidates or potential DILI cases, we often don't get the complete information with the narratives that's submitted. We see a lot of narratives which are pretty clear that they're machine generated, because there's no progression to the story, right? So you read it as a clinician and you clearly see that this is not something that's written by a clinical person who go from this initial sort of warning to then what investigations, what things are done. So it just reads very computer generated. And a lot of times the information gets really repeated and it's very redundant, and it's just very difficult to actually read those narratives. And then we also have these custom variables, which are to the present not really defined on how you submit this, right? There's no controlled terminology from CDISC on some of these custom variables that over time, they were not mentioned, for example, in the FDA DILI guidance, but we know since then that there are other new variables that may be helpful in the DILI assessment. And so let's talk about adverse event data, right? So symptoms of DILI can be very nonspecific. Sometimes it's just abdominal pain or abdominal discomfort, or jaundice, and it's not, or just nausea and vomiting. So there's, I very rarely ever see sponsors actually put drug-induced liver injury as a preferred term, although I've seen it once. But I would imagine if industry is putting that term, they are pretty certain that this has occurred. And in general, we tend to see other vague terms. So they're very non-specific, and I don't find the AE data to be very helpful. And the other thing we often see is that terms don't get reported unless they're considered to be clinical significant for labs. So for example, you may see a transaminase elevation, but it's not real in the labs, but it's not reported in the AE data set because it's considered by the investigator at that time to be not significant. So they don't report it. And then the other thing is that abnormal labs are really not reflected in the investigation SOC, right, in the AE data set. So we really, the AE data set is a very limited use when we're trying to identify this DILI as a potential signal. And so this is I'm going to mention a couple posters that we presented at the AASLD, which is the annual American Society for Study of Liver Disease. That's the big liver academic organization. So we presented this one. This was Dr. Lang's, who working with Skip, and presented this, where she looked at 22 market locations. She was the clinical reviewer on the DILI team, and she looked at 22 market applications that they were consulted on within the OND with regards to potential DILI issue, and she found a lot of inadequacies in the data, including missing subjects on the DILI screening plots, or also known as the EDISH. And then there's been really insufficient evaluation for non-DILI causes, right? Often we don't know what workup is done. And if the workup is, if the workup is not mentioned, is it because it was negative or was it just not done? The narratives are very incomplete. I already kind of elucidated to that earlier. And there was unclear or incomplete data on current medications, because concomitant medication is an important evaluation when you're looking at case level data. There's also really lack of sequential lab results over time. So again, making the adjudication process really challenging. And finally, there's also incomplete follow-up. So those are some of the challenges that was identified. And then this one was my ORIS fellow, and I looked at this, is we looked at liver biochemistry values from 119 clinical trials submitted to the FDA. We had a lot of filters, but we ended up looking at these trials that met our criteria, which is in adults, they had to have a comparative arm, they had to have complete ADLB, they had to have, a baseline versus subsequent value. So a lot of try to clean up the data. But if you look at the range of upper limits of normal that's been reported in these 119 trials, you can see that there is a huge range of the ULN reported for ALT. And if when we assess for DILI, we assess based on 3 times upper limits of normal, right? That's one of the criteria for Hy's Law. And if your ULN is 80, that's a huge number. 3 times 80 is 240, compared to if your ULN was only 15, right? So there is a lot of, there could be a lot of discrepancies between how you assess them. And then within a single trial, you can see the max number of unique ALT ULNs was 54. And that we found that 86% of the trials reported ALTs, they were greater than the standard 33, which is widely accepted by the hepatologist. And that's one that's used also for other liver diseases as well. And this is because we asked this, why is there such a huge range? I think that's a natural question. Well, it's because when the population, since Hy Zimmerman looked at these data to come up with the Hy's Law, the population in general has trended toward, as we know, more metabolic related sort of conditions such as obesity and fatty liver disease and all of these things. So our trends for upper limits of normal have been trending up in the population. And there's also intrinsic machine variabilities, although we think that that really can't account for this vast range, this huge range that we're seeing. But you can see that this is potentially problematic. And then when you use a different cutoff, you actually impact how many patients go into these quadrants. So basically, I think the bottom line is, there is really challenges in having standardized lab reports. And when there's missing data or data that's not fully reported, it really makes adjudicating for DILI even more challenging. And so common types of missing data, I think some of them have already been identified in the previous slide with Ling's poster, but we often get these missing baseline values where they're just not reported. So we don't know what the patient's baseline start with, and that can impact how you assess. Because liver labs are not always although in general, most clinical trials have a enrollment criteria for what their liver labs, but it's not always included. And sometimes that really becomes challenging when we don't know what the patient's baseline are. There's also lots of missing and incomplete labs, right? Because the common ones we get are AST, ALT, and total bilirubin. But we are also looking at other types of DILI, which includes cholestatic DILI or something that's autoimmune related, in which case you have additional labs that's required, and they're rarely pre-specified in the clinical trials as to be collected. And even if they are, a lot of the labs that reflect liver function, not just transaminase, but actual liver function, such as INR and platelets, they may not be routinely collected in this workup, right? And then the other thing is we have an issue with is sort of missing measurements. We'll see in the narrative, we've commonly seen this issue, which is challenging, is we'll take the ADLB and we will do the EDISH plot, and we'll see no patient in the right upper quadrant, right, in the Hy's Law quadrant. Then we'll get 5 narratives of potential DILI. And then we're wondering, well, why are we getting narratives of potential DILI when we don't see any of these patients in the Hy's Law plot? And so we're scratching our heads, and you read the narratives and you find that there's labs that meets Hy's Law criteria that's reported by the sponsor that doesn't show up in the ADLB. And that's really frustrating because now we're relying on the sponsor to tell us, to do their due diligence. And we like to think that they do, but obviously this is something that, we want to know. And so if it's not in the ADLB, it's difficult for us to do our own assessment. And we're really dependent on the sponsors to submit that information. And if, sponsors don't submit that information, then that information potentially could be lost, right? The other issues are like if the patients get submitted, a lot of times that lab is also not submitted. So the patient gets jaundice, they go to their ER. They don't go to the site for a visit, right? So all the labs that's in the EMR, they may not be extracted and reported to us. There's also data quality issues. Like I already mentioned this about the variability in the, and we also get these, when we did our initial lab cleanup, we found a lot of labs that were clearly erroneous, like they're incompatible with your life. If you're a clinician, you just know you cannot have a total bilirubin of greater than 1,000, right? And that's often maybe a unit conversion issue or something else, but that's just incompatible with your life when your normal bilirubin is 1.2. Right, so it should not be over 1,000, but we get these really clearly erroneous values that are reported, and that's not cleaned up doing the data quality check. And then we also have sponsors that submit like non-standard lab names, like they'll come up with additional variable names for ALT, or they'll use total bilirubin to reflect urine bilirubin instead of the standard like control terminologies that CDISC has. So these are some of the issues that I've personally encountered and in talking to the DILI team that we've seen in the submissions. So this is sort of what I think are possible reasons for DILI data deficiency, right? One, it is a rare occurrence. So there's very limited experience and expertise in how to adjudicate for this, right? And also the development rules are really quite stringent now, so that even development programs, as soon as they sort of get a hint for DILI, because they don't want to take the risk, a lot of times there is early termination. We also get a lot of, even though the trials do specify trial termination criteria or drug stopping criteria, sometimes the patients aren't even there yet. They make it to the 3 times ALT, and as we know, there is sometimes an adjustment, right, for the patients that are on drugs. Sometimes they can get the elevation in the transaminases, but then they don't actually progress to liver necrosis. So they can just make it into the Temple's corollary quadrant with the elevation in transaminase, but they never get the jaundice. They don't get the total bilirubin. But it's hard to tell if they are stopped on drug early, right? So sometimes sponsors get very nervous, and as soon as they see a rise in ALT to 3 times or 4 times, they're thinking, okay, this is the potential DILI. Even if the patient didn't have jaundice, they don't want to proceed. So they stop the patient on drug early. So we never know if the patient actually progresses to have DILI. So that really even makes it even more challenging because we have even less cases. And then there's really inconsistent practices in industry. And partly, I think the FDA really has not given a lot of guidance in this area. But, talking about, it wasn't until the very most recent update, and I'll have a slide on that talks about inclusion of sort of local labs or other study visits. And then the data elements, again, the technical specification we put out that even mentions some of these variable names is only really just over a year old. So a lot of this is not really consistently practiced in the industry. And then the wide range of normal evaluation of other causes is also inconsistent. I think that I've seen different pharmaceutical companies having different sort of DILI workup procedures, right? There's no widely accepted workup. And then finally, the narrative generation, again, that's also no consistent practice. Some companies large companies will use a machine probably to generate these narratives. Some companies use hepatologists. So some companies just use investigators who may or may not have any experience in DILI assessment. So there is a wide variability that contributes to these data issues. So what can sponsors do to get assessment with these submissions, right? So I think for a case level data that we would like, ideally, for sponsors to submit all laboratory values. We would also like a graphical profile and a good narrative. And I'll show you examples of what that necessarily entails. And so I want to draw your attention to this latest update in this last quarter in September to the TCG, that if you haven't noticed this language, this is the new language in here that we are asking 2 separate domains for lab results to be submitted. Because of the difference in SI and LB, we're asking that a separate domain called LC, and we're still working on what LC exactly stands for. But as far as for the SDTM domains, we're asking an LC be identical to LB, but in conventional units be submitted. And that ideally, we would like both of these units to come from the vendor rather than to have, sometimes I see, a column for sort of standardizing the lab where you're converting a lab originally in the SI into a conventional unit, when actually the source laboratory can report both values in the 2 different units. So this is the language that I took from the latest TCG update, if you aren't aware of this. Next slide. So this is an example of a line graph that Skip often uses. He's the DILI team lead that I stole this slide from. But he basically talks about how graphically you can display. And this is an example of a good graphical display, right? Where you see when the patient is on the study drug, right? So there's ideally a drug start, stop, or hold, or pause display is a temporal sort of timeline of the different elevations of the liver labs when you stop other concomitant medications that potentially can be a culprit and any other sort of outcomes included in here, right? So in here, you can see that there was a sharp rise in green is AST, and then you see that there was also a rise accompanied by a rise in ALT as well as bilirubin, right? So this is very helpful when you're adjudicating case level data. This is another graphical patient profile that a software may be able to generate, where you have the physics, you have the exposure, the disposition, and then any sort of AE terms that may accompany at the same time. And then you see sort of this trend in labs over time and concomitant medications start and stopping to help with the adjudication. Because what we often get is we'll get a tabular description of the labs, but then there's no sort of this type of graphical display is actually really, really helpful in the sense that you overlay all the events on the same timeline. And I think, this is truly where a picture is really much more useful than just the word narrative. Okay, and what should you include in the narrative? Well, if you are going to have your machine generated, I think it should just, for starters, just read well. I've read ones where it's really terrible and there's just repeated repetitions of the same data over and over again, right? But you need to describe good baseline data. What did the patient's baseline liver biochemistry profile look like? Did they have normal? Was it slightly elevated, right? Did they have any underlying pre-existing liver disease? We really need a well-documented time course, as I've already shown in the previous slide, about a temporal display of the data. Any signs and symptoms. And again, we know that this can be sometimes difficult because the signs and symptoms are generally pretty vague. And so they could be very nonspecific, right? But then the important thing here is evaluation for alternative causes, including workups for viral hepatitis. Now, sometimes trials already have a baseline workup of viral hepatitis, and we've had sponsors ask us, well, do we need to do it again? Yes, the answer is yes. Within reason, right? Obviously, if the patient's been vaccinated against or has been tested for baseline A, B, and C, you don't necessarily, you don't necessarily have to test them again. But something like hepatitis E is not often tested at baseline, and that's one that we're seeing an increasing incidence of patients having that. So you'd still need to reassess the need for whether you should test the patient for hepatitis and redo an evaluation of alcohol, because a lot of times we know that patients may not be, may develop, or situations may change and they may have increased exposure to alcohol. There's also, an evaluation should be conducted for biliary disease, right? Whether they have gallstones, any new concomitant medications, especially herbal medicine that's not necessarily reported, and any autoimmune hepatitis markers, and any other conditions, right, that may be relevant in here. And I'll show you an example later on of a table. So this is actually a table that's taken out that we send on our IRs very often to sponsors when we ask. And this is sort of just a table that you will also find in the technical specification for the MASH that talks about the AD-DILI custom data set. But essentially, these are some example tests that you might consider. And we're not saying that you have to do all of these, but it's helpful to know if it was even performed or not. And then we can, if you provide this data, then we know, okay, well, the sponsor did CMV, IgM, then we can go into the lab to look for that data, right? If you did a liver biopsy, we can go look into the data submitted to see what the histology was. It's challenging when we don't know what was done and what was not done. And so here's the link, and I put that and this is the link to the technical specification for NASH. And we put this guidance out about 2 years ago, and this is a level 2 guidance that is meant to be a companion to the therapeutic indication, to the level 1 guidance that DHN put out about drug development programs for NASH. And so this guidance contains how you should submit the data for these type of trials. But what we included in here is data submission guidances for workup of DILI. And so we added some custom flags in the ADAE and ADLB that includes like hepatic injury, potential DILI AE event. So these are things that can help us do the evaluation for DILI, right? And also baseline labs, because for a lot of these patients, especially for the patients in with underlying liver disease, they may start with abnormal labs. And during the course of the trial, if their labs improve, the DILI SMEs feels that you should have a new baseline now. If their labs improve to, say, normal, well, then when you assess for DILI, you should not be assessing it based on the baseline that they started in the trial, but a new baseline for purposes of evaluating DILI. And then there's also this R value, which is, again, it's it's something that hepatologists are aware of, that they do incorporate into current DILI adjudication. So a number of these variables are new that we have proposed to be included in the data submission. And this is an example of the ADAE where you can see some additional flags for hepatic injury, where we are trying to identify potential DILI events, right? And you can also create your own custom queries, but the idea is that we're trying to identify specific data that's important for evaluation of DILI. And again, this is an example of some specific flags that we've recommended to be included in the ADLB, where you have the baseline flag, which I mentioned before, and an onset where the sponsor thinks that the initial values is meeting the lab criteria for onset of DILI. And so the guidance also recommends a AD-DILI data set, which is a custom DILI data set, where we do provide some guidances, but if the sponsors have any questions or concerns, and some of the cutoffs are different depending on your trial room population, so we do recommend that the sponsors seek agreement with DHN. If you are, obviously if your application is related to a liver disease, but if it's not, you can still have your respective clinical divisions and talk to them about what should be, and they can always consult the DILI team on the DHN to come up with a reasonable criteria on what to use. The customer parameters, again, I showed you the table before, where we do add custom flags, potential outcomes, and even actions taken. And there are some custom variables, including the calculated R value, the baseline ratios. These are all some things that we use and look at in our adjudication. And then any sort of components of DILI workup that I showed you in the slides before with the table. So again, this is AD-DILI variables, where you can see that you can include these variables. Parameters could include potential DILI, outcome, action taken, and these are variables that you would include in the param, and then you could then add the more detailed information, but this is just an example. And we also include in the guidance additional SDTM domains that talks about where serology, or if you do biopsy, where things should be placed, to try to provide some guidance on how to best submit the data in the appropriate domains. And we also ask a reference to indicate the use of this tech spec in the TS domain. So these are some additional references. We are working to update the DILI guidance. And then when we do that, then hopefully this will include a lot of these other information in there. This is the CIOM's drug-induced DILI publication that they talk about how to assess for DILI. And here's the link to the tech spec that I referenced before about, even though it's for NASH, there's a significant amount of recommendations in this document for how to submit DILI-related data. And then I want to put a pitch in for safety planning, as that was sort of also one of the goals for that I saw in the opening slide that Susan shared, is that safety analysis really requires planning early in the development cycle. And by early, I mean, after the end of phase 2, where you're going into planning for your pivotal trials, you should be thinking about, if DILI was a potential identified AESI or adverse events of interest, you want to sort of take this into planning because of these additional data requirements. And so you can leverage the Type C optional meeting that should happen between your end of phase 2 and pre-NDA meeting. And I've included a link here that talks about leveraging these meetings during safety planning from the SBIA website. That there's a YouTube talk that I did on Type C meetings and how to leverage that. But this is a potential opportunity for sponsors to engage with their review divisions on how to submit data, and specifically to talk about any sort of data requirements for the market application submission. So these are everybody who helped to work on the the DILI working group, and just special thanks to everybody. And that's the end of my presentation. I'm going to stop sharing.
Thank you very much, Veronica. And now I'll share slides here to have some panel discussion from 3 of the members of our working group. We really appreciate your presentation.
Hi. This is Mike.
Hi, Mike.
Hello.
Go ahead, Mike.
Okay. So you may be wondering, why do I have a 15-year-old reference on a slide on missing data here? And I'd be wondering the same thing, because to me, some of the most ubiquitous and pragmatic advances in our field has been our approach to missing data, specifically the estimand framework, with the pre-specification of intercurrent events and the strategies for dealing with them when estimating statistics. And you think about filing an IND, we're expected to specify variables of interest, summary statistics, of safety events, and the estimation of confidence intervals. And the estimand framework, of course, is completely relevant to those kinds of efforts. However, if you think about the presentation that Veronica just gave us, we see the emphasis on looking at individual subject data. The EDISH plots themselves are subject level data. The subject level profiles are, of course, our subject level data, and the narratives of it's all subject level data. They're not statistics per se. So it's not a question of what kind of imputation method we're going to use when this data is missing. It's not that. I do think the idea of intercurrent events, or at least the idea of the reason capturing why things are missing, could be informative to the interpretation of this subject level data. And that's one of the reasons why this book is here, because it does emphasize the idea that you capture why something's missing. And another is that you follow subjects even after they've left treatment to collect information. But I think, and that leads into what I think are the key message covered in this book. And you can see the emphasis because it's covered in chapters 2 and chapters 3. And it's all about how to reduce the frequency of missing data, right? We can't eliminate missing data, but what can we do ahead of time to get as much as we can to ensure we have the right data? And this emphasis on the importance of how we collect safety data goes into trying to address some of the concerns that were expressed in the presentation. And it's also rolls into the next panelist discussion. So at this point, I'm going to pass it off to Jürgen to talk about the ASAP and its role in these issues and others.
Yeah, thank you, Mike, for the introduction, and thank you for Veronica also for mentioning the importance of planning earlier. So in fact, I think when thinking about specifically the missing data issue, there is one point I would like to address, and this is not missing individual data, but something I would call missing by design. There was the example in Veronica's slide that sometimes there's complete missing of baseline characteristics and baseline data. So this is probably something that hasn't been considered at the time when the trial was designed. So that's something I would call like missing by design. And these kind of issues, and also the questions as to what kind of data you need, what kind of additional information you need to collect, this is something you need to have on the radar screen fairly early in your trials, and therefore planning for that is pretty important. And the good news is you don't have to start from scratch to think about that. There is a paper on that from this working group here, and I've seen that Barbara was a lead author or is the lead author of the publications also online here. And that's basically on aggregate safety planning, assessment planning for the drug development life cycle. And what we came up there with is something we would consider to be a very flexible but also comprehensive framework for all aspects of safety planning. It's pretty much a proactive approach where you describe basically how you're going along over time. It completes basically also the point that you need to look at both aggregate data and patient level data, and we've seen how important the patient level data are specifically for DILI. It facilitates the interdisciplinary collaboration. And also, as you have seen, probably it requires here a lot of input, not only from clinical people, safety, from statistics, but also from people setting up the trials like data management and others. So one thing you could think of here, given the importance of the liver safety topic and the DILI topic, to have something like DILI as a safety topic of interest as a default in your clinical development programs to discuss it a little further. There's one on the next slide. Go there. There is an example for how the list of safety topics of interest could look like. Here you see something where liver tox is given as an example for a potential risk. So that's probably at a later stage, as Veronica was talking about. So at some point, you may have the information, may have some findings from preclinical. That's probably the point where you put something like liver tox on the list of potential risks. And here you have an opportunity to describe basically what you plan to do. And this overview is also very helpful. You have a documented decision in that place, and think of situation where team members change, you have new people coming on board. This gives you an opportunity to really document what has been decided, and you can make sure that what has been discussed and decided in the past gets really implemented in the future as well. I would like to point you also to the other safety topics of interest. So it's not only identified and potential risks that go into this list. You have the opportunity to also list other safety topics of interest. Here's an example of something that potentially could something like an anticipated event, because there's an increased risk in the study population, and you would like to see does your drug have an impact to further increase the risk in that team in that area. But you could also consider to have liver tox or DILI on that list at a very, very early stage already. So just to make sure it's on the radar of the team, and you look at that and in regular time intervals, and then take appropriate steps when moving ahead. Well, once you have done all that, and then collected all the data, we have seen a number of nice graphical presentations from Veronica, and I will hand over to the next panelist to maybe talk a little bit about that.
Yeah, thanks, Jürgen. So on this slide, if you imagine in the estimate framework, this is the estimator part. So once we have the purpose, the target of estimation, we develop the ISG group under this safety group, develop some tools to achieve the purpose, achieve the adjudication purpose, data adjudication purpose. So as you can see, the map shows the Hy's law plot. So it's interactive. This doesn't show up here, but there's animation when you click on the dots. So we'll show the animation over time. But also to Veronica's point, this allow the user to drill down to the RV showed and other lab values to adjudicate whether this is DILI or not DILI. So back to the earlier missing data point. So when we have missing data, then it's even more important to take into account the totality of the data, right? So if there may be other domains, AE or other comments that can fill the gap, can help us to recover the missing information. So that's why the patient profile is very important. So thanks to the ISG teams, Jeremy, Spencer, Preston, So we develop a patient safety profile app that serves similar purpose as Veronica showed earlier. So that the idea is the same. It's to overlay longitudinal domains on the same page so that we can make sense, piece together all the different components and make sense of the patient's journey. So you can open the link to learn more about these tools. They are all under this safety graphic ISG working group. And we really cherish the open source collaboration among industry and academia and regulators. So this represents a joint collaboration with all these different collaborators. But last but not least, I want to also make a connection to the missing data part. So as Mike said, the best solution mitigation is to prevent missing data, right? So so these two actually help the medical safety monitoring, right? And even data quality monitoring. So we actually, in my place, we use tools like this to monitor data quality, because you can quickly tell by looking at the trajectory which visit the patient has a missing data point. Then we should really go all in to query the data points and make sure we don't lose that. We don't have a missing data point, right? We are actually doing this for function data, efficacy data, but also we are using tools like this to check safety data, especially local labs. So I want to return this back to Susan.
Thank you, Xiao. Thank you, Mike, and thank you, Jürgen. We have some time now for some Q&A. So I know there were some comments in the chat. I see Aaron. Aaron had some questions. Aaron, would you be interested to come off mute and say more about your question? And
Sure. Can you hear
perhaps-
me okay?
Yes.
Sure. I have a question that goes back to something that Veronica had presented earlier regarding the different baseline measurements for specifically when you're measuring potential DILI cases. And I think she had mentioned how if a patient, for example, starts off with, let's say, potentially abnormal like a liver enzyme measurements, like you may want to have a second like a baseline measurement that's separate to this. I find this interesting and challenging in a number of respects, because most trials that I've seen, we usually measure baselines at very fixed points for pretty much all patients when we're looking at pretty much any measure from labs and others. And it raises a question about at what point would we want to be able to determine when a second baseline measure would take place? Because let's say hypothetically, I can think of an example where you would have a patient that would come in at the initial, say, inception of the trial. They would have abnormal readings in their say liver enzymes and such, which could indicate potential liver issues. But that measurement can change, but it may not shift to a what one would call a normal level, but it would be some of the enzyme levels may decrease. I mean, is there any sort of defined criteria and that's currently listed in the FDA or in other sort of discussions about at what point would we want to determine a second baseline endpoint? Has that been discussed at any point? I'm curious if perhaps anyone on the panel can perhaps speak to that.
So maybe I can start. So it's a very good question. And I would say that the short answer is no. There's no one single answer that can basically guide on how you identify a new baseline. A lot of this comes out of sort of, I think there's a lot of things to consider, right? So if your trial has to do with a patient that has an underlying liver disease, that and starts with an abnormal baseline. It also depends on the therapeutic indication. So for example, I'll use NASH as an example because I'm just more familiar with it with my work in that therapeutic area. But for example, in NASH, we know that there is a variable baseline. And when sponsors submit, if you look at the guidance itself, even to establish a baseline, there's variability, and we ask them to capture 2 and then try to take an average or have some sort of an approach that's pre-agreed with the division, the clinical division. And if it's another type of liver disease that's not NASH, for example, you may have a different pattern of baseline. So there are so many confounding factors that comes into establishing this baseline. What we're trying to create is this sense for sponsors to think that, hey, if my drug is working, my patient with underlying liver disease may actually get an improved, and I should be more judicious when we're assessing for DILI, because when we need to look at baseline. And this is something that's probably not something that you would capture per se as part of your standard labs for protocol, because we assume that you're going to be capturing liver labs, right, as part of your the labs that are captured as part of the protocol. But it's more of a after fact when you're having these cases adjudicated by a panel for those patients, you want to go back and specifically flag these sort of, if you see the patient's labs improve, you want to take that into consideration, because that's how we are going to adjudicate it when we look at it. Now, it's very hard to come up with a very specific individual criteria that's sort of a blanket statement that crosses everything, because I think every case is going to be a little bit different depending on your therapeutic area, depending on your patient population, and then depending on your drugs, whether DILI is a true adverse event of interest, right? So there's a lot of other factors in consideration. The main thing is to just identify that this is a potential thing that you want to think about, and then talk with the division that you're working with in saying, okay, well, is this something that you want us to do, and then work with the division to come up a reasonable criteria to establish that. Does that help answer your question, Aaron?
Yes, I believe so. Yes, thank you for pointing that out. I think it is important, to sort of have that early discussion early on to, in a sense, determine what are some of the, whether or not, as you say, like DILI should be considered an adverse event of special interest, and also considering, I guess, at different points, like what the patient population, what would be considered appropriate baseline measurements that you would want to take. So yeah, I think that's something that we have to address really at the protocol development level, I would imagine, as the protocols being developed, rather than sort of ad hoc, as I think might occasionally, I would assume others have probably seen.
Yeah, and while this is going to be more of an issue if you're developing drugs in the space of liver disease in general, right? Because you're assuming you're starting with the patient population, and while we know that they're not necessarily at increased risk of DILI per se, at least not hepatocellular DILI, their pre-existing liver disease may impact the metabolism of the drug itself, which can also indirectly increase the risk. So, and then the other thing is, so this is a very special case where I think depending on the population that you have to, again, I totally agree with discussing with the review division early.
Thank you.
Thanks for the question, Aaron, and for your thorough answer, Veronica. Are there others from the panel who have more they'd like to say on this?
Yeah, maybe to just add one aspect here. I mean, yes, the planning aspect at the study level is important, but I think we should not forget that planning level at the program level is even more important to achieve consistency and to put you into the position that at the end of the day, when you submit, basically, you can really tell a comprehensive story across all studies at the time of submission. And planning and the ASAP helps you with that as well.
Thanks, Jürgen. Let's see, there is, oh, nice to see you, Leela Lackey. Is there a document describing FDA's causality assessment approach? Leela, would you like to come on board and ask your question directly?
Can you hear me?
Yeah, hi.
Okay, great. I know, and I know this is probably not the main emphasis of the presentation, but like when there's suspected DILI, FDA goes through an approach to assess whether it is DILI and to what degree. And I know there are different levels of certainty that we can have. And I'm just wondering if anyone could point out a reference to that approach if it's publicly available.
Specific to the DILI, the CIOMS actually goes through the different types of like probable, that's what you're asking, right?
Okay, so it's the CIOMS reference?
CIOMS goes through that, and so that's one approach. We are also working to update our guidance for the DILI guidance, so we'll be discussing some of that in there as well. And I think the DILI-N also has, the DILI network also has some publications that you might want to consider also looking up. I think there's some publications that looks at causality assessment, but the CIOMS is very detailed about assessing DILI and all the considerations. It's like a huge document, but I highly recommend reading that as a reference if you want to learn more about causality assessment in there.
Okay, thanks, Veronica. I by no means will be doing causality assessments.
Yes.
I'm just trying to understand the results when I see them. So thank you.
Yeah, I mean, usually when DILI is a true concern, companies will often form a hepatic adjudication panel with experts in DILI to blindly assess these potential cases. The tricky part comes when I think, DILI signal was not a previously identified AESI, is only during safety assessment where it comes out as a potential issue, and then to hindsight, go back and look at the data, it becomes a lot more challenging. Yeah.
Mm-hmm.
Thank you for the question, Leila. With that, I believe we are out of time for the Q&A. So I just wanted to say a couple of words. First of all, thanks to everyone for coming to today's webinar. I see 52 people are on at this point. And thank you very much to Dr. Pei for your great presentation, and for our panelists, Mike Fries, Jürgen Kubler, and Xiao Ni for their great responses with respect to how this working group is reflecting on these kinds of things. I just wanted to make a comment on how important it is that we're working together across our respective disciplines of medicine and statistics and data science. They're all in, I think you can all see that they're all in play here, and really appreciate the importance of that collaboration and how that's playing out here. I also wanted to thank one person that hasn't been mentioned is Meng-Chun Lee. She is affiliated with Merck and has been a longtime member of this working group and started the concept of this webinar. So thank you, Meng-Chun, and for your work behind the scenes today. For next time, coming up soon, November 6th, we have a webinar on the impact of CIOMS MEDRA labeling grouping and AEGIS white paper recommendations on aggregate safety planning and assessments. Another of our physician members of this working group, Barbara Hendrickson, has been putting this together. So please join us then, and you'll be able to find the information for this coming through the same channel where you found this one. Any comments from from Veronica or any of the panelists as we complete today?
Nothing else from me. Thank you very much for the opportunity to share this with you.
Yes, thank you for sharing it with all of us, Veronica.
Thanks. It was a nice discussion.
Thank you, Mike,
Yeah.
for your contribution.
Thanks very much. Very much enjoyed it.
Thanks, Jürgen.
Same here. Thank you.
Thank you. Thank you,
Bye-bye
Susan.
now.
Thank you,
Bye.
everyone. Bye.
Bye.